Very good afternoon to one and all. Today, the topic of my oral presentation is transcriptomic analysis of Tavernera filifolia brotheli. I am Talibali Momin from Department of Botany, Faculty of Science, MS University, Baroda. So these are the images of Tavernera filifolia, the in flowering stage, and these are the root samples that has been extracted, and this is in its natural habitat. So Shastras has described two types of licorice, okay, that is Thalaja, that is terrestrial, and Jalaja, aquatic, wherein Thalaja have been described as Glycyrrhiza glabra. However, Tavernera quinifila has not been yet validated as Jalaja. There are some preliminary phytochemical verifications done by Zor and Mangalorkar. So germplasm of Tavernera quinifolia from Gujarat state was collected, okay, from Dinodar Forest, Kutch, then Manjuka village, Rajkot, and the Bagodra from Ahmedabad roadside. Among the three germplasm, root biomass was analyzed, and we obtained 40 to 280 gram of biomass along with the sucrose content as 47.89 microgram per ml and glycyrrhizin content, which is the primary main secondary metabolite component for our interest. It was obtained at 0.153 milligram per gram, and further analysis was carried out from these samples, which was obtained from the Bagodra area for phytochemical as well as molecular analysis. So Tavernera quirifolia is a branch structure under shrub belonging to family Fabaceae. It has the genus Tavernera has approximately 16 species, and it is owing to its sweet taste of root, it is also known as Indian licorice. Okay. So Glycyrrhiza glabra is popularly known as licorice, and then it is most popular medicinal plant is used in feed and food, and there are many uses of this plant in industries like food, herbal, cosmetic, and nutraceutical. However, in present scenario, Tavernera quinifolia has opened a new potential alternative for the commercially known Glycyrrhiza glabra. There are only few documentations that have been recorded for Tavernera quinifolia, and my primary focus work is on the transcriptomic or gene in this gene of these plants. There have been only 16 proteins that have been reported in the NCBI. So preliminary analysis was done for the glycyrrhizin, then liquidizine, glabridine, and liquidinine. These are the four main components that are being present in the Glycyrrhiza glabra. We are looking for the same components in our Tavernera quinifolia. However, the preliminary analysis done by Mangalorkar has shown 27 phytocomponents that are similar to Glycyrrhiza glabra. They are shown over here. So what is the main significance of Tavernera quinifolia as compared to the Glycyrrhiza glabra plant? The harvesting time is for 3 years for Glycyrrhiza glabra, and for Tavernera quinifolia it's 1 year. The soil use is acidic, and here it is saline. The root production is 3 tons per acre, and here it is 4 to 5 tons. However, the glycyrrhizin content is more in Glycyrrhiza glabra, but it can be used from Tavernera quinifolia as well. And we are not having any idea regarding the glabridine, and we are searching for that. This is the proposed pathway for biosynthesis of glycyrrhizin proposed by Seki et al. In 2011. They have shown this pathway in the Glycyrrhiza uralensis species, and among these, there are few enzymes, that is 2,3-oxidosqualene, then beta-amyrin, then 11-oxo-beta-amyrin, and 30-hydroxo-beta-amyrin. These all 4 genes we have got in our data. So here is the comparative analysis done with the Glycyrrhiza glabra extract fraction for the pure compound, and estimation and validation was done using the liquid chromatography along with mass spectra. and the standard glycyrrhizin was used. The solvent system was butanol, acetic acid, and water. Saturation time was approximately 30 minutes. These were the results. Calibration curve was obtained. The main molecular output was by doing the NGS analysis. We have run two runs for the Tavenera route, and we obtained approx 5.5 GB of data. Total sequence were around 2 crore 66 lakh 52,000, and the net average read length was 169. After doing and filtering all these raw reads, we obtained the final assembly of this sequence as 35,590 transcript. The total length was approx 1 crore 49 lakh 28,144. The maximum length of sequence was around 5,617 and N50 value was 441. Then the blast text was done against the non-redacted protein database, and further analysis were done with different tools. These are the tools that I have used for the de novo assembly and annotation. Blast to go was performed, and after that, for mapping and annotation, Blast to go tool was used. These are the project statistic of our analysis. Among the 35,590 transcripts, total sequences, we have got 27,884 with the blast sheet, 22,510 with the mapping, and 21,066 with the annotations. These are the tag distribution. Among the blasted 27,884, the maximum were shown by the gene ontology slave, and mapped were around 22,510. There were 7,706 sequences for which there was no blast. And among the blast is the top match was among the Medico Tranquillata. The blast hit showed maximum with the Medico Tranquillata, and the second was Cicer Arietemu. It was around 16,044 blast hits were from the total 35,590 sequences. All these plants belongs to the Fabaceae family, and the top hit species were among the Cicer Arietemu. Medico Tranquillata, they have shown the data over here, and all these species maximum belongs to the Fabaceae family. The mapping database source was from the Interprotein database, Uniprot database, GeoCentral, GeoC, and Ensemble plant database. This was the evidence code distribution for Blasties for the Tavenera plant. These are the gene ontology distribution by the level, showing the gene ontology for the biological process, molecular function, and cellular component. These are the gene ontology sequences that have been matched for the biological processes, shows the biosynthetic processes, cellular protein modification process as the maximum sequence. Then there were molecular functions, which included nucleotide binding and hydrolysis activities as maximum for the molecular function. And for cellular component, membrane and nucleus and cytoplasm were among the top gene ontologies match for the cellular component. The Kyoto Encyclopedia of Gene and Genome was carried out for my data, and there we obtained approx 496 QIDs were generated from 35,590 unigenes of the Tavenera plant, and among them 246 enzymes were reported first time from this plant, and many pathways and secondary metabolites were first reported from this plant from our data. The cis-terpenoid and triterpenoid pathway shows three genes, that is the FDFT1, that is farnesyl diphosphate, farnesyl transferase, LUP4, and PSM. These are the three genes that have been obtained from cis-terpenoid and triterpenoid biosynthesis pathway. Then three genes were obtained from terpenoid backbone biosynthesis. One gene was from the steroid biosynthesis. Three genes were again obtained from the carotenoid biosynthesis. One gene was from the flavonoid biosynthesis. Two genes were again obtained from the monoterpenoid biosynthesis. These are the UGT8, and these are the end products of the glycyrrhizin that is being produced from the Seki et al. pathway. Z18 biosynthesis. Enzyme code distribution was revealed from the blast hit, and among them, the hydrolysis type of enzymes were maximum. It includes more approx 3000 sequence hit was there, followed by transferases and oxidoreductase. These are the top 15 transcription factor families detected from the Tavernera cuneifolia root transcriptome, and among them, the top were WD40 like and C2H2. Thank you.